Okay, 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 okay. So this video is gonna be on the immortal jellyfish. Immortal jellyfish. For living forever. Living forever. So we're gonna focus on the immortal jellyfish. Um living forever. Because the you see, once you remove um basically the multi jellyfish living forever basically uh the multi jellyfish is literally a jellyfish that lives forever because it goes through the phases of its life, and it dies. It doesn't die. I would say it dies. It dies. It dies. I'll say it dies. But the people that dies are encoded into the parts of jellyfish, because I'm saying that the jellyfish dies, but being immortal, it has to compress, basically, it has to read the code. Basically, jellyfish is immortal because it takes a segment of its code. The code becomes compressed into the junk DNA. So it's reading and writing junk DNA, but it's not using as junk DNA. So let's call it DNA. Basically, the DNA that the jellyfish uses contains all of the genomes um, that can be expanded. So when you reproduce the genome, when it goes to a certain stage, it starts creating new type of proteins. And I'm saying, so you got the four proteins: adenine, thymine, cytosine, guanine. But you can have is it RNA or DNA? RNA or is it RNA or proteins? The Is it RNA that came first, or is it the proteins that came first? Because the proteins are the gene sequences. The RNA are function perform the phases of operations. So what I'm saying is the RNA, the proteins are fold. Proteins fold. They don't perform the operation, but they have the nucleotides. What am I saying? I'm trying to give you an idea of what this is going to. So, I have not really remembered my biology class, but I'm hypothesizing. I'm creating it in my best interest, so I can continue researching further. So, to live forever, you have to have the DNA, but DNA proteins fold, because the protein folded after the RNA has read it. So the RNA has to read the DNA. The RNA has to a reader. RNA reads the gene sequences, and then the proteins fold. So in that aspect, you have something that a compressed. You have a compressed gene, and when you expand the genome, when you expand the genomes, the gene sequences, you then have the organism back to where the start of the initial state of the organism, which is very important to understand, because the initial phase of the organism. So that's the idea. So you have that's what I'm saying. That's how one way to live forever, because the DNA can be compressed, not like but to compress it means, you have to fold it. You have to because the protein, basically, to compress it means to fold it. So you have to fold it. Basically, the how what type of folders? So you have the folding algorithms. I'll define them as topology, the way they are folded, because you have protein folding, which is an NP complete problem. See, what I'm saying, what I'm trying to hint at is that the proteins fold, and because they can be read while the fold, because they can be read, um, to my best knowledge, while they're folded, after they're folded, you know, you have something that can represent the same. It's basically like basically you have so if you have like it's a color coding algorithm. So you have if you have a color of red pixels RGBA, Like R red, green, RGGB, BGR. So you have one read, one red, two greens, uh uh two blues. I don't like using I don't like comparing to computer code, but I'm giving my understanding of it. So it's not computer code I'm inferring to. I'm thinking about the Jenkins structure itself, just to let you know. So you have one R, two greens, two blues, one green, and one red. So one green. So you see basically I'm saying that this is not incomplete, by the way. So I'm saying that um the representation can be compressed, can be folded. So you can keep folding instruction set because the instruction set represents the simplest form of the proteins when they fold. What's protein folding called? Is it a representation? Basically, I can't remember the sequences. Of how proteins fold. So, no one knows how proteins fold for now, yeah, but someone will figure it out, let me just find it out. How does protein folding work? Because if I remember this is the only thing I'm struggling with. It's a physical proportion floating a ribosome amino acid chain from unstable random coils into a more three-dimensional structure. Yeah, so this is what I'm saying. So alpha helix, B sheet, quaternary structures, beta sheet. So we have. I'm looking at this diagram here. So well, people know how protein. Is it well understood how protein folding work? Protein folding is the physical process in which a protein, after synthesis by a ribosome, as a linear chain of amino acids, changed from an unstable random coil into a more ordered three-dimensional structure. Yeah. So yeah. So basically, I'm saying that the R yeah. So this I'm saying. Yeah. So DNA is alpha helix, spiral formation is DNA. Now I'm reading about it. Driving force supporting for. Let me just show my screen. Share my screen. Yeah, yeah. So we have it here. So what I say? Protein folding, as is read, can be unwound. So when you unwind the gene, you get you can RNA the functions because you want to maintain the RNA structure of the genome that has been read. So the functions remain the RNA, which is the reader and writer genomes, when they're read, to reproduce the same amino acid at exactly the same phase, so to assemble, which assembles into a structure. That's how you live forever. But that structure being compressed means that it can perform more operations, perform more functions. Yeah, more functions. Yeah, that's what I mean. More functions. So that's the idea. So um, yeah. So you have there's nothing as a junk DNA. There's no because the DNA that in your body has been encoded. It has been encoded. It has been encoded. So it has been it's its own category of DNA. So it's biologically its own DNA. But to live for the years to go through a phase. So the phase is like if I want to compress myself to my consciousness, for saying consciousness, I don't like that analogy. It's not biological. If I want to compress, if I want to represent um all the colors of dominant and recessive genes of the eye, color. My eye color is brown. My eye is brown right now. Is it black? Well, I can't tell. Well, let's say it's brown. So I think my hand was brown. So I come basically, I get one. So I get the dominant phenotype. But once that starts interacting at a certain phase, I get a different phenotype. I so the pheno so my phenotype is brown, which is basically the physical construct. Now my genotype is all of the colors, or all the shades of color that can represent brown, and that is and that's the genome. Basically, the sequence of the genome is the nucleotide itself, which are the let's call it composites. So you have an RNA which dealing the functions, which I don't really know how the functions work, functions, but the from the process of the phenotype. But you have the composite which is genotype, which is the DNA. Yeah, which is so you get phenotype and genotype. So if I want to represent if I want to represent my color of my eye, the color is brown, I take only two colors, gray. How to make brown? How to make brown? So you take all the functions of brown. I have to remember this because I'm building my own model of it. How to make brown? If you just say black, brown color, orange and blue. So I take all the genes can give me orange and blue, but I don't have it like color. So let's just say red and green equals brown. So red and green equals brown. And so that'll be a complementary color. So two opposite colors on the color wheel, which are composites, which are complementary. So two complementary colors are red and green equals brown. So I can do that with computer program too. So let's just say, but no one has red eye in the real world. No one has red eye. But I'll use an example. So red and green equals brown. So let's say someone has a red eye, God forbid, and that person is green eye. So to separate, so that means the phenotype of me having a brown eye would have to include one red and one RG R or G the responsible genotype that encode one phenotype. And when that's encoded, I have the DNA, which can be used, which are the functions, the features, the functions. Uh, so I need to get the amino acids, yeah, and it has to be compressed. So it has to be basically it has to be unwound. So I'm saying that the DNA has to be unwound because you have to fold it to have to go through the reverse process. Because the when you want to live when how the jellyfish does it when it's how's the motor because it goes through a phase and that phase can be reversible. So it's not reversible. So it loses. It's not reversible. It's irreversible because he's losing a complete phenotype. It loses a complete phenotype of that cellular organism. So it cannot basically. So I'm saying, I'm hypothesizing jellyfish cannot undo the process. Because it's a one-way process to be transformed into a new creature. So it dies. So it dies because only the brain remains. And that brain encodes all the neurological genome and phenome that will responsible for recreating that organism like a brand as a brand new organism. Thank you for watching this video.